Hello there, welcome to the world of Succinyl, where your academic needs are met. Today we are going to continue from where we stopped from our previous lecture, Porphyrins. So, what will be the properties for these porphyrins? What are the unique features of porphyrins? One of them is that they have the ability to combine with metals. The metals, for example, include iron, magnesium, and other ones too. The porphyrins can occur, tetrapyrroles can occur in different aspects of nature, and these include chlorophyll, myoglobin, hemoglobin, cytochrome P450. The reason I mention all of this is because they are quite important in different aspects of life, including muscle contraction, transport of oxygen and carbon dioxide, as seen in heme, as well as cytochromes. Then also in the activity of xenobiotic transformation enzymes like cytochrome P450. What are they trying to talk and say? These porphyrins, they feel combined with metals. The metals they include, they feel like iron, they feel be magnesium or other ones, depending on what they try to form, and they live for different parts of this life where you even know safe. So the examples where these porphyrins occur as now in form of heme, hemoglobin, myoglobin, cytochromes, we had to show you for years. So after this, we'll go right into a very, very important derivative of porphyrin or tetrapyrroles, which is heme. What is heme? Remember, the porphyrins include chlorophyll as well, and chlorophyll is useful in photosynthesis. And in that case, it is a form of porphyrin that contains magnesium and not iron. So heme is an important derivative of porphyrin. Heme is an iron-containing porphyrin that basically function as a prosthetic group in different enzymes and proteins. What does prosthetic mean? Prosthetic means that it is tightly attached to the proteins or enzymes in such a way that once detached, it will destroy the enzyme or the protein. Which I decided to talk and say, heme, it contains iron, will be magnesium or take note. And now prosthetic group really found for proteins and enzymes. What I mean I say prosthetic group means say if you come out the heme from where it forms, such as protein or enzyme, it will destroy and it will cast the enzyme or the protein, that kind of thing. So moving on, the heme, they're useful for different parts of life, such as the ones that they found in hemoglobin, myoglobin, cytochromes, such as cytochrome P450, and those ones that they found for the electron transport chain. And these guys now, they help for different aspects, such as the one they found for the red blood cells, for oxygen and so CO2 transport, also for the muscle, for cellular respiration, same as that's for proper function of the muscles, so I can feel they run up and down. Then the cytochromes, what they found for the inner membrane of the mitochondria, they help for generation of ATP. Say mitochondria are powers of the cell, what they cause and these cytochromes, they help do this thing, they generate energy. And for xenobiotic metabolism, means they help destroy those drugs. We say they take those supposed to be drug addicts. How the body they help and put this cytochrome P450 they found inside the liver. So what am I simply saying? I'm simply saying that the porphyrin can occur as heme, and this heme is usually found in different macromolecules such as hemoglobin, myoglobin, cytochromes, either cytochrome P450 or the ones found in the electron transport chain. The ones found in the myoglobin is useful in cellular respiration for muscular activities to enable cellular respiration during muscle activity. Then also the red blood cells for the effective transport of oxygen as well as carbon dioxide. And the xenobiotics, transbiotic transformation, cytochrome P450 found in the liver helps to destroy the toxic xenobiotics consumed in form of drugs, as illustrated here. Drugs, as illustrated by the diagram here, showing the drugs, as drugs can be biotransformed by the liver with the aid of cytochrome P450 enzymes. Now we'll look at the biosynthesis of heme. How is heme produced by the body? How is heme produced for the body? First things first, all cells can produce heme, but there are certain cells of the body that have this special responsibility of producing heme. There are two major ones. They are the erythroid cells of the bone marrow and the hepatocytes of the liver. But majority of the heme produced by the body is by the bone marrow cells, that's the erythroid cells of the bone marrow. But the little percentage, which is 15%, is by the hepatocytes. Take note. And 85% is produced by the erythroid cells of the bone marrow. Take note of that, please. And moving on, what kind of cells also can produce this kind of this heme, they include the reticulocytes. Please take note, the reticulocytes. Reticulocytes are just immature red blood cells, young red blood cells can produce heme as well. These young red blood cells, they are called reticulocytes, as seen here. But mind you, not all red blood cells can produce heme. Take note, you remember that from our knowledge of biology that red blood cells can live as long as 120 days. After 120 days, they are dead, they are destroyed, they are eliminated from the body. Take note, the young red blood cells, reticulocytes, are the ones that are able to produce heme, because from as illustrated here, they have the organelles to produce heme. You understand? So, but the mature red blood cells, they are devoid of organelles, or they are devoid of nucleus, so they don't have the ability to produce heme. Take note of this, as illustrated here. So this is a clear distinction between cells, the kind of red blood cells that can produce heme and those that cannot. So let's move on to the biosynthesis of the biochemistry, or the pathway involved in the biosynthesis of heme. Okay, let's delve into the biosynthesis proper. We cannot talk about the biosynthesis of a particular compound without looking at the pathway. So let's look at the pathway. So we'll look how the pathway be. Now, the pathway of heme biosynthesis shortens between two organelles It shortens between two organelles. The two organelles, or two parts or compartments of the cell, the two compartments include the matrix of the mitochondria and the cytosol. The process starts from the matrix of the mitochondria and also ends in the matrix of the mitochondria, but some intermediates are found in the cytosol. The reason for this is because the enzymes are crucial for this process are found in these two compartments of the cell. Therefore, these two compartments of the cell are useful in the biosynthesis of heme. What they try to talk now, say the biosynthesis they happen for two parts of the cell. They happen for the cytosol, they happen for the mitochondria, the matrix of inside the mitochondria, and then the matrix. The reason I say the enzymes they help for this process, they occur for those two compartments. The precursors are important because they are different for two different kind of organisms, for animals or fungi, and also for plants. For plants, it is crucial because the precursors are unique. For plants, it's a different precursor. For animals, they are different precursors. For animals, they include glycine and succinyl-CoA. Succinyl-CoA is extracted from T-cell cycle that happens in the same compartment of the cell, which is the matrix of the mitochondria. Then it forms ALA. ALA means δ-aminolevulinic acid. The enzyme there is ALA synthase, and the coenzyme is PLP. PLP is a derivative of vitamin B6. So in this process, it is a condensation reaction, and it forms ALA, which is δ-aminolevulinic acid. This is a rate limiting step, and this helps to form the ALA, and the ALA is transported out of the matrix of mitochondria into the cytosol, where it can be used for the subsequent steps. The steps include the conversion to form Porphobilinogen. Porphobilinogen is an important precursor because it is the first pyrrole. Take note, the first pyrrole. And obviously, if you check the structure in the next part, we'll discover that the structures have connections, they have relations. We'll look at it shortly. This Porphobilinogen is used to form the next tetrapyrrole, but it's not cyclic, it's a linear tetrapyrrole, which is called hydroxymethylbilane. The linear tetrapyrrole, and this can be used to form the next intermediate, which is Porphyrin 3. The enzymes here for these steps include ALE dehydratase, Porphyrin 1 synthase, Porphyrin 3 co-synthase. Please take note that in this process, there can be a spontaneous conversion of hydroxymethylbilane to form Porphyrin 1. Why Porphyrin 1 is irrelevant biochemically or biologically, so therefore can cause harmful situations that will be looked at in a subsequent video. Porphyria. So looking at this, these enzymes, they have their relevance. The dehydratase means that there's a removal of water. So that enzyme helps to extract water or remove water. The Porphyrin 1 synthase helps to remove ammonium ions in form of amino groups. So the next one, so moving on, the Porphyrin 3 is converted to Porphyrin 3 with the aid of the enzyme Porphyrin decarboxylase. That's a decarbonylation step, not just a decarbonylation, it's an oxidative decarbonylation, but we'll see shortly. Then there's the conversion of Porphyrin 3 to protoporphyrin 3 by the enzyme Porphyrin oxidase. Same oxidative decarbonylation process. And the next step is the oxidation process aided by protoporphyrin oxidase to form protoporphyrin 3. Then the last one is an incorporation step whereby the metal is incorporated, which is the ion is incorporated to form heme. Which I talk about since morning. I say, whoa, this step or this process of heme biosynthesis, it involves two compartments, what I don't talk before, and they involve glycine and succinyl-CoA. Succinyl-CoA come from Synthase cycle. Remember, Synthase cycle get succinyl-CoA inside the process. To form, for my name, succinyl-CoA, is the day for Synthase cycle to form ALE. And the ALE, now important step. The formation of ALE is important step because na rate limiting step. They help control the entire pathway. So for my name, succinyl-CoA. So the ALE they use until they form the next steps. The ALE they come out for the cytosol enter, so from the mitochondrial matrix enter the cytosol, and this ALE we can combine two of them. We can combine to form Porphobilinogen. If you observe and well, that step now dehydration step of the step because water they come for that the enzyme then ALE dehydratase. Then you can form Porphobilinogen. Porphobilinogen will convert to form hydroxymethylbilin. That Porphobilinogen now the first pyrrole, which are the enzyme the heme na Tetrapyrrole. So you can convert again. Because you need 4, so you need 4 of these pyrrole to form Tetrapyrrole. Therefore, the first Tetrapyrrole they form for the process now hydroxymethylbilin, but not linear. They straight, but you don't say what they come back with the local capacity and they circular form, so they just circularize. You know they help circularize this thing and the Porphyrinogen 3 co-synthase. So the Porphyrinogen 3 is converted to, or they converted to Porphyrinogen 3. That enzyme at oxidative decarbonization step. So carbon 4 side they come out inside, then you can go inside the matrix of the mitochondria again, can continue the process. That enzyme they convert the Porphyrinogen 3 to form Porphyrin 3. Now, Porphyrin oxidase. That step is still oxidative decarbonization step. I'll show you the next part. Then here, you can form Porphyrin 3 by the enzyme Porphyrin oxidase. Then finally, when the main catalyst enter, which is the ion, when enter the form the final product, which is heme. Take note. So the next thing here they show say, okay, you don't talk about this, say no put structures. I know they explain kind of structure, the structure they combine, they join, they show how they join, they form the differences we need to form. So if you observe how you go see how they join, how they come out the different chemicals for inside, the different functional groups for inside. So take note. If you observe how you go see the succinyl-CoA they form from there and they join to call glycine to constantly form δ-aminolevulinic acid. If you call δ-aminolevulinic acid, if you call 5-aminolevulinic acid, delta later of the Greek letter. So, but now no concern, not just the observer as they go. So at this point, this is a point where you get to understand the structural features of the pathway, how they shuttle between one point of the cell to the other point of the cell. So if you look at it now, the linear Tetrapyrrole is illustrated here as seen, and the first pyrrole is also illustrated there, obviously, and how it is cyclized. It is the removal of water that makes it cyclize. If you observe it at that point, you see it, you realize that it's a very important process or step in the biosynthesis process. Then there is the formation of Porphyrin 3, which is an oxidative decarbonization step. That's oxidative decarbonization step, which is illustrated there as well. Then this converts to the Porphyrin 3, then to Porphyrin 3, then finally to the final intermediate. So if you observe, you can say all the chemicals that they come out. You observe the functional group they come out, the water, the CO2, the exact part of the compound where they happen. They this pathway to explain. So take note, I beg you. So at this junction, we'll round off this talk. This would conclude and say that so far so good. Let's try and see if we've learned something from what we have just said or what I've just learned. So there are some certain questions I can use. You can pause the video to go over the question and see if you have actually learned something. So these questions are to bring you up to speed as to different parts of the pathway and also different parts of this lecture. So thank you very much. God bless you. I remain fascinated, and you can like and follow, and you will not regret the next video that will come. Thank you. God bless you.